Well, good afternoon. And I've missed you so much. It's just given me the blues. So that was a nice little opportunity there just to get the blues out of my system. So today we're going to be looking at brain scanning. So let me just share my screen with you and pull up the appropriate bit. And here it is. Okay, so let's go from the beginning. Okay, so as science has developed over the years and become more sophisticated, psychologists have suddenly become able to look at what's going on inside the heads of people as they're doing behaviors, which is a major step forward, as we'll see in a minute. So we need to understand how brain scanning works and what are the pluses and minuses of that process, okay? So you should be able to explain what we mean by brain scans by the end of this. You should know two different types of brain scanning method and be able to explain them. And you should be able to explain the strengths and weaknesses of each of those methods. Okay, so that's the overall aims of the next, I don't know, it should be about 10 minutes, hopefully. Okay, so thinking about brain scans, this is the board's definition here at the top, and I've added a little bit on. Okay, so it's a research method that involves taking images of the living brain, and that's an important thing, to investigate brain function. So we've got things like PET scans, fMRI scans, CAT scans. Now, as far as the exam goes, and in terms of limiting what we need to study, the two that I'm going to focus on are the ones that you need to know about are MRI scans and PET scans. So those are the two. These ones are PET scans. And this is an MRI machine here, and we'll see more about that in a moment. Okay, so up until the 1950s, which isn't all that long ago, up until the 1950s, if we wanted to know what was wrong with someone's brain, by actually, which physically wrong with someone's brain, we wouldn't be able to find out until they died, really. So for example, Paul Broca, who was interested in language and so forth, he would basically make a note of the kind of behaviors and problems that his patients would have, and then he'd be able to figure out what he thought the reasons might be after the person had died. So he would cut their brains open and check it and say, oh yeah, that bit there is a particularly strange shape or whatever. That's probably a reason. And by doing those kind of things, he actually found, as I think we covered earlier in the start of the biological approach, that he actually discovered an area that's named after him that's responsible for language. Okay, so trouble with that approach, of course, is that if we have to wait until someone's died to find out what's causing their problem, it's not really going to help that person all that much. Okay, so in the 1950s, we got a major step forward when people were able to actually invest effort and get a picture of what was going on inside the brain while someone's still alive. And the first machine that they came up with was the electroencephalograph, which is a machine that involves putting electrodes on the outside of your scalp. So as we can see, that person with the cap on there in the picture, each of those terminals and so forth is an electrode, and you can measure what's going on in how much electricity is being used in each of those particular parts of the brain. Okay, so Dement and Kleitman did a really famous study and made a discovery that if people were wearing this when they were asleep, Dement and Kleitman found that when they went into rapid eye movement sleep, when they went into that particular stage, there was also a lot going on in the brain that suggested that people were dreaming at that time. So they were able to make quite a good connection between rapid eye movement sleep and dreaming. The trouble is, though, it's based on, if you like, it's like listening in from the outside. And it's not as precise as more modern methods that we've got. So I'm going to talk about a couple of those now. So we've got MRI scans. Those are some pictures there at the top of that one. Magnetic resonance imaging. Now, how that works is that someone lies down in one of those big tubey machines. Very strong electric magnetic current is passed through them. And what that does is that changes the direction, or it realigns all the cells in their body. And then the magnet is switched off. And the moment the magnet is switched off, all the cells go back to where they were before. So they change back in direction. But as they change, they give off radio waves. And what the computer in a MRI scanner does is turns all of those radio signals into a picture of what the brain would actually look like, which is quite incredible, isn't it? So that's how it works. It uses radio signals to create a picture of the brain. Functional MRI is the same process, only that they take pictures while someone in the tube is actually doing something. So they might ask someone to do some maths calculations, for example, and then they would look at what was happening in the brain at that stage. Okay. Maguire et al., a good example of the use of MRI scanning was Maguire et al. They found out that taxi drivers had a bigger hippocampus, or two of them, so bigger hippocampi than non-taxi drivers. And their findings actually helped us come to the understanding that the hippocampus is involved in spatial memory. So when you're thinking about, the route that you take home, for example, or the route that you would take to school if you were allowed to go to school at the moment, that you'd be using your hippocampus to think about those things. Okay. So that's how MRI scans work, and an example, Maguire et al. And then we have PET scans. You'll be more familiar with this because we did this with Rain et al., as I'll say in a minute. So in PET scans, they use radioactive glucose. Now, the brain uses glucose when it's active. So if a piece of the brain is very active, a section of the brain is active when you're doing a task, that will be using a lot of glucose. Now, if we put radioactive glucose into the body and then use a scanner that picks that up, then what will happen is that we will see the most active parts of the brain will be most strongly highlighted. And in the picture above there, we can see that the really active parts here are in red. That shows that there's a lot of glucose activity going on in those bits, and then much less in the blue and the green bits. Okay, so Rain et al., that's a good example. That's something that we looked at in component 1. They use this method to tell the difference, or to see if there are any physical differences in the brains of murderers and non-murderers. And they found there were, but as we know, they were also very cautious about drawing any causal conclusions. Just because we've got a particular shape of brain of one sort or another, that doesn't mean to say that we will definitely go on to kill someone. Right. Okay, so those are the two types of scans. Now let's think about what are the advantages and disadvantages of them. Okay, so looking at MRI scans first, what they do that's very useful is that it gives a very good picture of soft brain tissue compared to something like a CAT scan. That's one that we haven't looked at, and you don't really need to look at all of these different methods. But a CAT scan is one that's really based on X-rays. So it gives us a more accurate and detailed image of the brain than a CAT scan. And also, as we don't have to use a CAT scan, that means we don't have to expose people to potentially harmful X-rays. Okay, so that's two main advantages, and we use those advantages by comparing it to a different method. Looking at PET scans now, because PET scans are actually tracking glucose going around, they're actually revealing chemical information about the brain that MRI scans can't. There's a mistake on the PowerPoint, I'll change that. So that's useful. So that's telling us about chemical information, and then they can tell the difference between, for example, benign and malignant tumors, between tumors that are dangerous and ones that aren't. And also they show us, because they're showing the glucose activity in the brain, they're actually showing us the brain in action, which is better than what we would see in CAT scans, for example. Going to be downsides, of course. So think about MRI scans. If you think about the long tube that people have to go into, they can take a long time inside those tubes, and they can be quite uncomfortable. It's very noisy, and it can feel slightly claustrophobic. So that would be a problem for people who don't like confined spaces. PET scans, on the other hand, one of the key issues about them is that they're incredibly expensive to run when you're thinking about buying the radioactive glucose and all that kind of stuff. And so it's not easy for psychologists to use in research because it's quite expensive to get hold of. You're exposing patients to potentially harmful radioactive substance. So you can only use it a few times, really, otherwise it would start to become kind of dangerous. And also, as you can see from these two pictures here, it's much less precise than MRI scanning. If you look at this, you can see clearly the structures of the brain and the various bits. Over here, it's a bit more like an oil painting or something like that. Highly trained scientists obviously can interpret those, but the MRI scans are definitely making it a bit easier for them. So over to you. So for your next assignment then is to look at using this textbook in a presentation. You need to be able to describe two methods of brain scanning. That's two lots of 4 marks. So that's 5 minutes for each method for describing it. And then you need to outline one strength and one weakness of each brain scan, of each type, one strength and one weakness of brain scanning. So you can actually choose one method and do it like that. Okay. So if you can get that in for me for next week, that would be lovely. So I'll just finish off as we started with the blues theme tune for brain scanning. And goodbye, everyone.